Okay.
Yes, we
And thanks
can.
so much for inviting me to speak today. I'm really excited to talk to you guys a little bit about icy moons and the habitability therein. Um, and particularly with a focus of trying to couple observations with geology and chemistry and try to see how we can work through that chain from what we see, what we infer physically, chemically, and geologically in order to unveil hidden habitats that may be present on icy worlds far out in the outer solar system. Um, and to that end, the broad link that I'm trying to draw in this talk is to define habitability in a quantitative sense, firstly, and determine exactly the kind of physicochemical properties that we're looking for, then link that to the more fundamental chemistry and geology that needs to be ongoing in these requirements in order to sustain and enable planetary habitability. And then after that, we'll do a very quick overview of the observational suites on spacecraft that have been or are in transit to icy moons in the outer solar system and try and tease out how those observations work back down this flowchart to give us inferences about habitability. But the first thing that we really want to ask ourselves when we ask that question is, what exactly is a habitat anyway? What does an environment actually really need to offer in terms of the raw physics and chemistry that's ongoing to be considered suitable for life? Now, arguably, the most complete formal definition of habitability is a suite of conditions which is suitable for a known organism to survive and grow, whereby the most reliable assessments of that are based explicitly on empirical knowledge, so things we've seen in the field or things in the laboratory. Now we can reduce this thing down into a series of physical and chemical controls on biology, which is popularly shown in a kind of Venn diagram like this, where critically there's an additional time element associated as well. So we need all of these things through time in order to really sustain habitability long term and have the best chance of life being present in these environments. But the next question is, how do we quantify these things really and link them to a physical system, right? So in this case, what is energy? What does energy actually mean? I come from a physics background, so energy is really locked up in everything, locked up in mass coming from suns, etc. And what are these limits when we say environment and when it comes to pH, salinity, temperature? So shown in this way, it's quite vague, but we're going to run through each of these four things and try and quantify exactly what's needed in order to really come up with a thorough habitability assessment. The first one is pretty straightforward. You require a solvent um, in order to have the chemistry be active. On Earth, all the solvent that life uses is liquid water, so that's pretty easy for us to tick off straight away. We just have to look for liquid water. Energy is a little bit more abstract, so let's look at that in slightly more detail. Now, you may have seen when you read through the literature reference to a free energy or this property delta G to support arguments about habitability. And widely, it's also used in biogeosciences to work out energy fluxes for biology in the environment. So it's pretty well established. What this property tells us is how much energy is released by a chemical reaction at a given composition, temperature, and pressure. And a simple example of this is if you have a cat in a box with some kind of supply of fish, as the cat eats the fish, breaking down the complex chemistry within them to simpler compounds, it harvests the energy that gets released, and that's what keeps the cat alive. And this was famously described by Schrodinger back in the 40s as biology in a way feeding on negative entropy. But the thing is, like biology also grows. So physicists may then go on to wonder why this can happen, because it introduces additional complexity to the system. And the answer for why this works is because as long as the total entropy in the system increases, so you can build more complex stuff, provided that overall you're making the chemistry around you less complex. So in this scenario, the fish have been broken down and the cats have been able to grow. And this is a thermodynamic exploit that life is able to use in order to grow into very, very complicated systems. Given enough time, though, our cats encounter a new kind of problem. Once they've eaten all of their fish and they're surrounded only by bones, then there's no more energy available in this box that the cats are adapted to extract. In other words, the box is no longer habitable for our cats, unless they get some kind of energy resupply, which we would parameterize as a power, just simply energy provided per unit time. So this is that time element coming into the energy portion. Now that intro was quite abstract and quite oversimplified, but it does characterize the key thermodynamics of what biology does. Mechanistically, it realistically gets its energy by pushing electrons around, which is why another often discussed requirement you'll see in the literature is suitable redox chemistry, and you may often see species referred to as electron donors and electron acceptors. The very simplest organisms, the ones that we're most interested in the context of icy moons and astrobiology in general, have energy sources that are very simple chemical reactions, where biology catalyzes the movement of electrons between different chemical species, using a system known as an electron transport chain, which looks a little bit like this, depending on the life form that you're looking at. So that's energy taken care of. Life essentially needs a direct energy source, which could come through the source of through photons or through some kind of chemical interaction. And in it, for the chemical case, it gets that by breaking energy down. It then uses that energy to build new biomass and keep existing biomass healthy, which brings us to the next requirement, which is nutrients. So let's go back and look at our cats again. We glossed over a very important part, which is whether the fish actually contain the necessary chemicals to build more cats. So the key elements typically considered for building biomass are referred to as CHNOPS elements, standing for carbon, hydrogen, oxygen, phosphorus, and sulfur, with additional trace elements such as some transition metals, perhaps, depending on exactly which biological species we're looking at and its requirements. Now, in the case of our cats, they're probably fine. It's biology eating biology, so there's probably plenty of CHNOPS in those fishes. But for microbial species that feed on rocks instead, it's critical that those rocks contain the necessary nutrients and that they are, through some mechanism, made bioavailable. So this could slowly happen through time with chemical interaction, or it could be mediated by the biology itself. And this is an important hurdle to overcome on icy worlds like Enceladus. We'll come back to that slightly later, but provided you have a rock with the necessary nutrients and mechanisms to get them out, then we can satisfy this requirement. So that's energy and nutrients, and we've even talked about time a little bit already. But there's one more quite cryptic notch on this Venn diagram, which is the environment. Environment here refers to the survival criterion, essentially asking the question, is the energy and nutrients you're getting enough to hold yourself together against the local physical-chemical extremes where you find yourself? This can also be described as a power, and you may see this referred to as the maintenance power or the power demand in the environment. And this essentially needs to be smaller than the power supply we defined earlier in order to ensure survival. Otherwise, you may not be surviving for very long. We can think of this maintenance power as kind of the rate of energy input an organism needs to use to keep its biomechanical structure in check. And it varies depending on what the adverse conditions in the environment are. For instance, if you're in an environment that's slightly hotter than you're used to, your enzymes may have a suboptimal function, so you may not quite get as good a power supply, or they may simply decay more rapidly, so you'll need additional resources to keep on repairing them at a faster rate. One of the problems with maintenance power, though, is actually measuring it. And usually the estimated requirements in the literature span over several orders of magnitude, as we show in this figure. Let's look, for example, at temperature, where the maintenance trend, as you increase temperature, in any case, is probably exponential or so. But the baseline theoretical minimum estimates down here are 3 or 4 orders of magnitude lower than what has been derived in the laboratory. So realistically, the actual maintenance power for a typical species is going to be somewhere between these two values. Nonetheless, one thing that we can do to make a habitability assessment is to simply plot a power supply against the power demand on this figure. There we go. And see when one overtakes the other. So in this example, the power supply, which I've completely made up, is lower than the lab-derived maintenance power up until we get to about 340 Kelvin, where it starts to beat it out. So this species may, for example, be a thermophile and likes warm temperatures. Now, these sorts of functions can be drawn for different physicochemical properties, and we could even draw them in like 2D maps and 3D maps to show different survival limits affected by different key physicochemical parameters. But of course, all of those have quite significant uncertainties. Another complication is that, critically, everything I've talked about so far is organism specific. And by that, we mean that different organisms are going to have different adaptations, which changes both the energy supply that they get from the environment and the maintenance power that they require in an array of different settings. Nonetheless, the diversity of life is really vast and has worked out how to tolerate a really wide variety of extremes, which have been compiled by several authors over the years. For example, here on the right, we're showing a map that encompasses known survival combinations of polyextremophiles at different pHs, salinities, and temperatures. And this was made back in 2013, so that map's probably pushed a little wider now. So we know that life is diverse, but actually quantifying species-specific things remains quite difficult. So that completes the set and has hopefully demystified this Venn diagram quite a bit, at least in terms of how we can quantify all of these things. To quickly summarize, to ensure habitability, life needs a solvent, a robust energy source, which is usually in the form of free energy, liberated by chemistry or photons from solar power. It also needs an environment which is not so extreme that survival requires more energy than is available, and it requires a supply of nutrients to rebuild itself and build new biomass. Most of these things are all made available by local physical and chemical parameters, so things that we can constrain abiotically. And just finally, before we move on, a special note again to this time requirement, which has come up a few times already, and I just can't stress how important it is to have all of these things for a long period of time, not just a short, instantaneous burst of energy, because you wouldn't be able to maintain things for very long. Okay, so that's habitats. And we've already come quite a ways in the discussion so far to linking habitability to physicochemical properties. So let's have a quick look at how geology and chemistry, particularly in an icy world context, enables these habitability criterions to be met. And this will be quite qualitative, because hopefully you guys know quite a lot about icy moons already. So icy moons in the outer solar system typically have an ice-covered crust with an ocean of liquid water below, which may or may not interact with a rocky core at the bottom. And this figure is showing an example of different processes that may be ongoing in Enceladus's ocean. Now, depending on the moon itself, the ocean could connect to the surface, like it appears to do so for Enceladus, in the form of geysers that erupt from the south polar terrain. Although I should clarify that the jury is still out on the extent of the connectivity with this plume and the ocean, if it's connected at all. Another ongoing process that may liberate energy on icy moons is at the bottom of the oceans and is through rock-water interaction, which is very likely going on in Enceladus and possibly Europa as well. But it's not a guaranteed process on all icy moons. For instance, Titan likely has a high-pressure ice layer at its seafloor, which largely prevents extensive rock-water interaction in the present day, not to preclude it in the past. But incidentally, it's also difficult at the surface of Titan to interact with the ocean because the crust is just so thick and cold that it's difficult to fully traverse so far. Ice-ocean interaction and freezing melting cycles may also be of interest for habitability. So this is at the top of the oceans, because it's one way to mix fluids and have fluids constantly mixing to generate that critical, albeit probably quite small, energy gradient that life needs to survive. And so finally, we can ask ourselves whether these are really good habitat candidates. And for Enceladus in particular, we think that these might be, because it does have some degree of each of these things I just outlined. And we have much more data to back up what's going on Enceladus from the Cassini mission. So we're going to come back to Enceladus several times through the talk as a case study going forward. Now, that said, we need to quantitatively determine whether these can be good habitat candidates. And as we discussed in the first part, we can do that as long as they're sustained through time and they can provide energy, nutrients, and nice chemistry. All should be good for life, right? So this flowchart shows one way in which you could try to quantify habitability from geochemical insight. If we have some kind of mechanism to calculate the water chemistry in our proposed habitat, so we can get a free energy availability, couple that with nutrient availability in order to calculate a power supply available for life, compare that to a maintenance power required for life based on observations on the Earth, we can check whether one is larger than the other and test whether it's a good habitat candidate. Okay, so first let's have a quick qualitative look at the options we have on icy moons and analogs that may exist on Earth, because we require those analogs to get the specific biokinetic parameters for the biology we want to test. Rock water habitats that might be similar to icy moons do exist on the Earth. In the deep ocean, hydrothermal vent systems are erupting from the seafloor along fault lines and host a really diverse array of life and a slew of tasty energy gradients that biology makes use of. These occur as hot water flushes through rock systems and liberates energy and nutrients. And then as it bursts into the cooler seawater, it mixes and diversifies the chemistry that's ongoing. Similarly, we can also think more theoretically. And in theory and in the laboratory, we can test hypotheses about miniature habitats, like we talked a bit about earlier, where rocks provide most of what is needed. And then geophysical, geochemical activity might just be needed in order to help make it a little bit more bioavailable or deliver it to different places. On the other hand, ice, water, slushy habitats do also exist on Earth, although there are more caveats with the analogs to these. One example could be at the bases of glaciers, although there are an important difference with at the top of the ocean on an icy moon and with the bottom of the glacier on Earth is that on Earth, most of the carbon that enters that food chain is originating from photosynthesis, which is something that's really not very likely to be possible deep inside the interiors of icy moons in the outer solar system. Alternative analogs might include lakes deep below the subarctic or subantarctic. For example, shown here on the right is a lake some 4 kilometers or so below Antarctica. But both of these locations are kind of less well studied and have much more meager and slow energy supplies as compared to the rock-hosted ecosystems we just looked at. So now that we've established what a habitat is, we've set up some hypotheses about the settings that might be habitable on icy moons. And just to recap, the key things that we need are some kind of sustained chemical disequilibria, but at the same time, nice enough chemistry to keep yourself alive. Next, we need to ask ourselves the question, how do we deduce what's going on in these settings to make these real quantitative inferences about habitability using the observed data from these worlds? Well, given the structures that we just looked at, with oceans that are hidden deep below thick ice sheets, we can't simply go and sample these places and just scoop up some ocean water to see what's going on there. So we're left mostly to remote sensing observations. So let's have a very quick, brief look at the kit, and we'll see what it has told us in the past and may tell us in the future about these worlds. First, let's just quickly recap a few exciting spacecraft that have happened recently or are happening very soon. So on the left here, we have the Cassini mission, which really catapulted icy moon astrobiology over the past couple decades, exploring the Saturn system, and was responsible for some fundamental Titan science, alongside discovering Enceladus's surface geysers and the ocean itself, and giving us quite a lot of information about what may be going on in that ocean, amongst, of course, far more other planetary science that's not necessarily astrobiology related. The next generation of spacecraft recently launched, including Europa Clipper and JUICE, which are heading to the Jovian system. And then a little bit later down the line, hopefully Dragonfly is going to fly to Titan within the next decade or so as well. And it's not shown here, but there's also ESA's more recently announced flagship return to Enceladus a little bit further down the line. So there's a lot of things in the pipeline to be heading to icy moons soon. So it would be useful if theory people like me could understand what the information that they're going to tell us. Speaking of which, if you're a theory, hypothetical guy like me, computational guy, who knows very little about engineering, when you compile together the observing suite of spacecraft, you will end up with something like this, which is a slew of acronyms. And for our topic and for my background, this doesn't immediately tell me very much. None of these say really fancy things like life detector or habitability interferometer or redox sensor, right? But we do need to understand exactly what these things do tell us to make the scientific inferences about habitability we talked about earlier. So I'm going to hopefully try and demystify some of this by lumping these instruments into two broad categories, what they tell us about the geophysics and what they can tell us about chemistry. So any engineers in the audience, maybe mute your laptop for a few minutes because you may find what I'm going to say highly offensive, and please challenge me in the questions and answers session because I'm sure you know a lot more about this than I do. And also just one final preface, I'm focusing on habitability here. There's obviously so much other awesome science that these things can do, but we're going to focus specifically on the habitability question to keep things short. So our first broad category is geophysical observables. And the first easiest one to describe is simply optical imaging, or looking in the visible spectrum and just observing. So for example, the Cassini imaging science subsystem was responsible for many of the really magnificent images of things in the Saturn system that we now see in textbooks. But it also helps tease out some quite interesting surface topographical features. Like, for example, these are some boulders that are present on the around the south pole of Enceladus, and we're still not really sure why those boulders are there or how they were formed. But using imaging, we can also observe at different wavelengths to get additional information about the surface features of icy worlds. So here's an image of Enceladus' south pole, which was created by compiling data together using Cassini's VIMS instrument, which observed in the visible and in the infrared wavelengths. The area in red that we see here, which is now famously referred to as the tiger stripes, is indicating regions that were recently resurfaced and have a slightly different composition from the equatorial region, for example. And Cassini actually had a couple of infrared spectrometers. The other one focused on emission at longer wavelengths, which gives us slightly more information about the temperature profile. And overlapping with this region in the red here are temperatures that are about 100 Kelvin warmer than the surrounding surface. So this is already giving us some inklings about geophysical processes and temperature gradients at the surface. And an additional type of observation is to look at LiDAR or radar, which I've kind of lumped together here because they're mostly about topography mapping of surfaces. LiDAR works by rapidly firing lasers and then measuring the time that it requires for that light to get reflected back. And shown in this figure is the reflectance of Mars, showing the topography of its surface using data from the Mars Global Surveyor from about 20 years ago or so, just to give an idea of the specificity that LiDAR can give us. Radar works in a similar way by reflecting radio waves, and this is how Cassini was able to map hydrocarbon lakes and seas on the surface of Titan through its very, very thick, hazy atmosphere. But another use for radar that wasn't possible on Cassini, but may be possible with the upcoming Europa missions, is radar sounding, where radar waves are sent with the intention of penetrating the ice and reflecting back information from the subsurface. The hope is that this will help us probe the interior and map the ice-ocean interface of these worlds, and may also characterize things like the distribution of salts and the extent to which the ocean and the ice interact with each other. Finally, magnetometers are instruments that measure the strength and the direction of the magnetic fields as the spacecraft move around within them. Now, this can not only tell us a lot about the interior of the host planets, for instance, Saturn, Jupiter, and their magnetospheres, but it can also infer some information about their satellites. For instance, the magnetometer that was aboard Cassini observed very subtle changes in the magnetic field around Enceladus' south pole, which in the end turned out to be due to the water vapor that's erupting from its surface. Magnetometers can also provide information about the extent and the composition of subsurface oceans, if it's possible to observe small induced magnetic fields caused by the conducting salty water within these worlds. So that's remote geophysical sensing. If we are very lucky, we can also do some chemical observations, which I've lumped into two categories here. The first is spectroscopy, which overlaps quite a lot with some of the geophysical observations I just described, in particular looking at the absorption and emission spectra from the surfaces of these planets can tell us what chemical species may be present. For example, instruments aboard Clipper and aboard JUICE are hoping to tell us what salts could make up, for instance, these slightly different colored portions of Europa's surface, and maybe give us some information on how long they might have been there. The other type of chemical observation is mass spectrometry. If we're lucky enough to have plumes, as we do on Enceladus, mass spectrometers can help constrain exactly what chemicals are present within those plumes. Cassini had a couple of separate instruments. One was the ion and neutral mass spectrometer, which was specifically for volatile species in the space plume. And the other was the cosmic dust analyzer, which was able to analyze particles, larger particles like ice grains. And using these two instruments together, it was possible to estimate the composition of the ice and gas that erupts from Enceladus' south pole and identify some species which may originate from the ocean or even originate as far down as in high temperature rock-water interaction. And shown here is an example of one spectrum coming out of the INMS system. So that completes the full set. Although you may have noticed that links in this chain still feel a little bit blurry and vague, it's quite difficult how we get from the very abstract ideas of habitability all the way up to the observable features at the top still. But it's a process to tie all of these things together, right? And I hope that at the very least, this has given an idea of the pathway we can take to make those assessments and the uncertainties that we have to think about along the way. So to that end, before we finish this talk, I think we're really well primed for a rapid fire case study to see what Cassini observations can tell us about the likelihood of habitats being present on Enceladus. So throughout the talk, we've already covered most of the clues. So for the last few minutes, we're going to try and piece them together to make estimates about Enceladus' habitability. First, let's very quickly recap the relevant observations. So an ocean was found on Enceladus with plumes observed flowing out of the south polar terrain, which has been quite recently resurfaced and has a quite interesting heat signature. Cassini then flew through this plume with its mass spectrometer pointed at the plume source and analyzed the volatile makeup of the gases that are escaping into space. And then its cosmic dust analyzer observed ice grains that are thought to originate from these plumes and helped constrain the key salts that make up their source. These things together were then used to infer the composition of Enceladus' seawater, which incidentally is one of the first steps on our habitability modeling framework. So let's quickly revisit that. We need to relate this to the chemistry of a specific candidate environment and see if it ticks off the key criteria that we defined earlier: energy, nutrients, power supply, power demand, etc. Now, there are still necessarily lots of assumptions in performing this analysis, right? Are the chemical observations directly connected to the ocean? Are they diluted or concentrated in this transit? What is the temperature at the seafloor if that's the habitat we're looking at? These are all very much still open questions in Enceladus science today. Nonetheless, we can look at what has been inferred recently. So over the course of several flybys, these chemical species in the top right were determined to be, and many others, were determined to be present in Enceladus' spacecraft. It so happens that this chemical reaction is an ancient metabolism on the Earth known as hydrogenotrophic methanogenesis. Now, as models of the ocean have matured and slowly become better constrained, we've started to better understand the ocean composition, its pH, and its temperature, which is mostly pretty cold with possible hotspots below the seafloor. Nutrient availability does continue to be a bit of an open question, so for this example, we're going to leave it out for now. But what this means is that with some uncertainty, it has been possible to estimate the free energy available from this metabolism in Enceladus' bulk ocean. And I'm showing here the first estimate from the Wade et al. Paper in 2017, which first reported the hydrogen in the plume. But this has since been interpreted in several different ways by the authors below. And regardless, in most cases, this energy does seem to yield bioavailable energy in Enceladus-like conditions. So it seems energy yielding, but is it actually habitable? Let's look back at our flowchart again. This only tells us the very first step. To make the next leap, we need to decide whether this energy is kinetically accessible for life and whether the power supply it can generate is enough to outweigh the power demand associated with survival. Because remember, we're going to set aside nutrients for now. And so to finish us off, I'm just going to show how Enceladus' habitability space looks to me when we sweep across the uncertainties of its physicochemical conditions informed by the Cassini observations. We see essentially a spread of 3 different habitability scenarios. The first is quite straightforward. There's a chance that the seawater itself is uninhabitable for methanogens on Earth as we understand them. Although energy may be available to them, it's not kinetically accessible enough to really sustain a biosphere for an appreciable amount of time, or in some cases, it's actually not even enough to beat out the expected maintenance power in those settings. This probability varies with the local pH and temperature as shown here. So at high pHs, slightly less habitable, but at higher temperatures, it does seem to be getting slightly more habitable. The second scenario is that the ocean is habitable, or the seawater is habitable, but growth is primarily limited by the energy availability in that water. You know, of all the things that could be slowing a hypothetical biosphere down in this setting, energy seems to be the main one. So our assumption that to not think about nutrients and some other things is probably reasonable in this scenario. Finally, the third scenario is quite intriguing, and that it's the ocean is habitable, but doesn't seem to be limited by the energy available. What I mean by that is the ocean appears to be a buffet, so much so that it's almost suspicious how much food there seems to be available there on the table. And this tells us that there's likely something missing in our habitat model preventing growth, like missing nutrients, lack of space, some kind of additional toxicity, or of course, the null hypothesis, which is Enceladus is simply not inhabited at all. Or I suppose there's an additional option, which is our seawater models are just have too wide uncertainties, and so these scenarios aren't even present. Now, the spread of possibilities on Enceladus spans all 3 of these things. So even with all of the Cassini observations, there's still much more to constrain to truly understand the extent of Enceladus' habitability. And maybe we can talk in the Q&A, but lots of assumptions went into building this model, right? So we're still not hugely certain about these probabilities in these 3 scenarios. But to capture it in a sentence, we need to go back and we need to do more, get more data to find out more information. So that's all I had to share today. I think if I was to try and summarize everything, which is a very broad presentation, but if I was to try and put it into one sentence, I would say that observations and resulting geophysical chemical models, constraining things like tides, temperature, redox fluxes, disequilibria, etc., can help us constrain dynamic habitability in a quantitative way. And although we didn't talk about it today, we can also use that to constrain the size of local habitats and the size of any expected biomass that may be present using very similar properties. But it's very important to remember that uncertainties propagate through this process and are very important to characterize in order to test hypotheses about habitability. And with that, I'm more than happy to take any questions. And if we don't have time for all, then you're welcome to send me an email anytime. Thanks so much, everybody.